evening, everybody. Welcome to tonight's Tutor to You session of
photosynthesis, and we're going to split it up into the two main areas. So we've got the light dependent reaction, and we'll have two separate games on those for our sessions 1 and 2 games, and then we will transfers into the light independent reaction, have two little looks at that, and then we're going to finish on the limiting factors. So let's kick it all off with the light dependent stage, and we're going to start off with some nice multi-choice questions, so you can answer in the chat, everyone. So first question: where does the light dependent stage take place? So put your answers on the chat. Is it A, the stroma? Is it B, the outer chloroplast membrane? Is it C, the thylakoid membranes, or D, the cristae? So put it on the chat. Let's see what you have to say. What do you reckon for that good starter? Okie dokie. I can see that is going to be a nice one to start off. Yes, yeah. See, you're all on the right chapter page for that one. Right. So let's have a look at the answer. Let's see what we've got. It is, of course, the thylakoid membranes. Those chloroplast membranes are inside the chloroplast, making up the grana. That is exactly where the thylakoid membranes are, and that is where the light dependent stage takes place. Right, let's do another one. That's a good starter. Okie dokie. What happens after the primary pigment, that's chlorophyll A, absorbs energy that excites electrons? So we've got chlorophyll absorbing energy. The electrons are excited. What happens next? That's our next question. So do put it on the chat. Chlorophyll is photoionized.
We've just lost Ellena for a temporary second. Tracy, are you happy to take over?
Yeah, absolutely. Let's move on to the next one, guys. Let's have a look what we've got here. So number 3, as electrons move through the ETC, that's our electron transport chain, what happens next? What is this process about? Have a look at those 4 options there and see what you think. You have to read these ones quite carefully, I think. So look at the language quite carefully for this one. Have a little think for a minute. If you can, pop it in the chat. Have a little guess. Okay, so let's have a look. See if you got this one right. Let's have a look. Yeah, so hopefully you recognize that we don't ever want to lose electrons, do we? Energy, I should say. We don't want, so we would never choose B because that will offend the laws of physics. So energy is released is a much better way of putting that, using that language there for that one. So energy is released to pump those protons through the thylakoid membrane to create that proton gradient. So well done, you guys, for spotting that. We talked about that lovely phrase, energy is released, in our last week's live stream on respiration. So if you have a little look at that, and we take you through some of the language that those examiners really want to see on your exam papers. Okay, so I think we've got Ellena back now for the next one.
Yay, I'm back again. Okay, so next one. What is the role of NADP in the light-dependent reaction? Is it A, the final proton acceptor? Is it B, the final electron acceptor? C, it becomes reduced when hydrogen ions bind? Or is D, a proton carrier? Lots of bear traps there. Which one is it? Which one is it going to be? Put your on the chat, see what I'm up with. Okie dokie, what do we got? Yeah, I can see that it's this is another tough one, and it is, of course, B. So it's the final electron acceptor. It can't be a proton acceptor because protons don't have electrons, they're just hydrogen ions. So if it's going to be reduced, it becomes reduced NADP, it has to have atoms to do that. It can't have the ions. So C is the same way of saying the same thing. It's hydrogen ions binding to it. And of course, hydrogen ions are known as protons, so it can't be D. So that's why we've ended up with B being the correct one. So just watch out for that. It's a very common error. Okie doke, let's do the last of the multi-choice questions. So we've got the photolysis of water juices Which is the correct combination of those? Is it only protons and electrons? Is it oxygen, proton, electrons, or oxygen, electrons, just electrons? Which combination is correct for those? And let's have a look. It is just, of course, it's all of them. It's oxygen, that's going to be a waste product, protons, which are great for building up gradient for the chemiosmosis, and the will go off and fill the gap in one of the photosystems so that photophosphorylation can carry on. Well done, everyone. So let's move to the second part of our light dependent stage. Tracy, over to you. It's time for a trapdoor.
Yeah, so this is a nice one here. So you've got some choices, haven't you? And what we want to know is which of those events that you can see actually happen in the light dependent stage, and which ones have we just put in to be really sneaky and to try and fool you. Okay, so we're just thinking about the light dependent reaction here. Okay, so have a little look. It could be any number of ones that we've chosen, and hopefully you can apply that lovely process of elimination, that great exam technique, to sort of think, no, that can't possibly be right. So just let you have a little ponder there for a second. Don't forget to put your ones in the chat. Yeah, absolutely.
Yeah.
If you think, no,
go
that's
A1 or C3 or whatever,
or
just
type
use
out
the
the
letter
word
and the
you
number.
want. So let's have a look. Got quite a few red herrings put in there. Some of you might have already noticed. So just give you a little moment longer. There's quite a bit to take in there. Some of them will sound sort of right, and that's what you often get in exam questions, isn't it? Especially multi-choice ones. They put sort of sneaky sneakers in that are kind of half right. So did you spot them? So let's just give you another 10 seconds or so, and then we'll reveal the answers. Okay, so let's have a look. What have we got going on in the light dependent stage? Well, we have got some reduced NADP production. So this is an easy one to get confused because you kind of think, well, I think there's some NADs, but are they NADPs going on? Remember, your NADs are your coenzymes involved in respiration, but for photosynthesis, remember that starts with a P. Okay, and that's an easy way to remember it. You've got your NADPs going on, haven't you? You've got your reduced NADP being produced. So that's a nice, easy way of remembering that NADP is photosynthesis, your NADs and reduced NADs are produced in respiration. So hopefully you got that in that first column A. Then in B, we've got a couple, haven't we? So we've got our chemiosmosis taking place, which again is that similar thing to respiration happening here, isn't it? But we've got, instead of oxidative phosphorylation, we've got photophosphorylation going on, haven't we? Because it's all to do with light. Remember, we're in the light dependent stage, not the independent stage. Okay, so hopefully you guys sort of saw those two, thought,
Okie doke. So for light independent stage, we've got you a 3-2-1. It's one of our favourite games. You need to find 3 intermediates of the Calvin cycle. Intermediates are the substances which are made in the actual cycle itself. So 3 intermediates of the Calvin cycle. I'd like you to find 2 products of the light dependent reaction that are needed to reduce GP in this light independent reaction, the Calvin cycle. And there's just one named enzyme that fixes CO2. What is that one? You got 90 seconds. Let's see how you get on. "90 seconds is a long time, or is it not long enough?" This is a big question.
hopefully, some of the stuff we already talked about, you've got the juices flowing a bit now. So we're in the zone now, aren't we? We're fully into the Calvin cycle bit now. See, what did we produce in the light dependent stage? Because we just talked about that as well. So hopefully some of you have got some of those sort of things stuck in your head from the last game too.
Okay, right. Let's look at the 4 intermediates for the Calvin cycle. So, of course, we have RUBP, that's ribulose bisphosphate, and you do need to know its full name as well, especially AQA students, because they don't like you to reduce down to abbreviations. And then we have GP, which is glycerate 3 phosphate. So, again, get that one right. It's not glucose phosphate, it's glycerate 3 phosphate. And then we've got TP, triose phosphate. That one always has to be written out in full in an AQA exam. They're a bit more lenient on that one in OCR. Then we have got the 2 products of the light dependent stage that are used to reduce GP in the light independent stage, and that's ATP and reduced NADP. And then finally, our little enzyme we're after, of course, it's Rubisco. So that sets us going on our Calvin cycle and light independent stage. Over to you, Tracy.
All right, so here we've got a lovely gap fill. Who doesn't like a gap fill? I know I do. So if we have a look and see the text that we've got coming up. So here we've got a bit of a couple of paragraphs for you to read and try and get these gaps. And we will be revealing them in a minute as we sort of go along. So we're thinking about that intermediates, or one of the intermediates of the Calvin cycle, that can be used to make some really useful organic products. Okay, so we're at the real nitty gritty useful end of this whole process now. We've got our triose phosphate, which again, you AQA lot absolutely need to write that out in full. Don't put TP, you will lose marks, okay? We've got our starting material for lots of different things. So we've got here, it says starting material to make carbohydrates, lipids, and amino acids. Okay, great. And then what have we got? Well, we've been a bit sneaky here, but actually, this is a great example of a real sort of synoptic exam question. So it's not just about photosynthesis, is it? You've got to use your knowledge from biological molecules, perhaps from some transport parts. So, you know, you've really got to sort of think about other areas of the specification here. So just give you a few more sort of seconds just to have a little read through, and then we will have a little reveal in a minute. So just be quiet for a second while you have a good think. If you think you know what goes in the gap, pop it in the chat, please. We'd love to see what you're thinking tonight. All right, guys, so let's have a little read through then. So we've got our TP as a starting material to make carbs, lipids, and amino acids. So glucose reacts with, what's our first gap? Let's have a look. Fructose to make sucrose. Of course, this is right from biological molecules, that carbohydrate topic. Do you know your disaccharides, basically? Okay, so if you weren't sure about that one, have a little revise, because very, very popular exam question, that one. Okay, so really brush up on your disaccharides, I suggest. Let's move on. So sucrose moves through the plant by what? We'll clearly want to process. Did you get the process name? It's that sneaky one, that translocation. It's the one we tend to prefer transpiration. A lot of students, they think, oh, translocation, that's the one that I'm not so sure about. But just be aware that that is all about the movement of sucrose. It's that really important product from photosynthesis. What happens after? So we're moving that by translocation to cells that are going to use it as a respiratory substrate. And then we've got our triose phosphate, our TP, can be converted into glycerol, which reacts with, what you get, that double gap there. That was a big clue, wasn't it? That we needed 2 words. So fatty acids, okay? So now we're thinking about lipids, weren't we? To make triglycerides, and these are stored as oil droplets. If more glucose is made than needed, it can be converted to starch and stored as starch grains in the chloroplast, of course. When new plant cells are made, some glucose is used to make, what do we need here? Yep, cellulose for making the cell wall. Well done if you got that one. The amino acids made are vital for making new proteins. This does require a nitrogen source. Like what? What actually is it? It's nitrate ions in the stool. And a little tip here, don't talk about nitrogen being in the soil. Okay, so yes, we need nitrogen to make the amino acids, but the form it will take in the soil is in the form of nitrate. So make sure you realize that. Okay, don't be writing nitrogen in the soil, because we won't be giving you any marks. All right, well done for that one, and let's move on to our final activity.
So we have a connection spinner to finish off today's session with, and this is all to do with limiting factors. So you will know that there are several limiting factors that can limit the rate of photosynthesis. So what we've got to think for this is, what is the effect on the rate of photosynthesis when some of these things happen? Let's see where it lands. Okay, so what is the effect on the rate of photosynthesis of... Now, where's it going to go? And it's slowing down, I can see. And yes, today we are going to look at decreasing the light intensity. So if you decrease the light intensity, what effect does that have on the rate of photosynthesis? Now, you can just to squeeze them in the box, you could give me increases, you could say it decreases, or maybe just go with stops if that's what you think. There are three ones. Place your bets now, pop them in the chat. What's it going to be? One of those three. Okie dokie. Right, let's see what, if you were right, let's see what the answer is. So, of course, it decreases. The photosynthesis rate is less. If there's less light energy being absorbed by pigments like chlorophyll, there's going to be less light-dependent reaction. That means less ATP and reduced NADP are made, and therefore the rate of photosynthesis will be slower. Right, let's have another spin. Okay, what else are we going to be given today? Could be any of these. Oh, I don't know. Oh, it's water stress. That's a bit of a tricky one. Have a think about that. So suddenly there's water stress. What effect will that have on the rate of photosynthesis? Is it going to increase it? Is it going to decrease it? Or is it going to stop it? I wonder why. Let's have a think. Put your answers on the chat and let's see what we've got going on there. Okay, you are right. Yes, the rate is going to decrease and actually it will stop, because the thing about water stress is the stomata will close and that will prevent the CO2 from diffusing in through the stomata, off to the palisade cells, and then into the chloroplasts, into the stroma to do fixation. Well, if there's no eventually CO2 coming into the stroma, there'll be no fixation of CO2 with RUBP in the Calvin cycle, and actually the rate of photosynthesis will stop. So well done. That was a good, bold one to finish on. And thank you very much for coming along tonight for our second biology live stream ever. Oh, Laura, we can't hear you.
I always do that. Sorry. It has to happen to one of us, doesn't it?
Yeah.
So thank you, Tracy and Ellena. You've had a great session there on photosynthesis. Lots of tips have come through. If you want to watch this back on demand, you can. No doubt you'll need to, because you've covered lots of great and complicated stuff in there as well. It's like another language, biology. So absolutely fantastic session. Thank you to everybody for joining us. Thank you to Tracy and Ellena. And I'll be back next week with another session.
We've got a little bit of a break, so we've got a 2-week break, and then we're back on Wednesday slot. So see you in a couple of weeks' time.
Okay, fabulous. Look out for us on the Tutor to You website and Instagram, and we will see you very soon. Thank you very much. Good night, everybody.
Bye.